So before going into that, I just want to tell you that there are many kinds of mouse models. We can add genes, and that's called transgenic animals. So we express the protein there. We add a gene into the animal. We can delete or alter a gene. That's called knockout. We can put in the gene in a knockout. That's called knock-in. And we can do chromosome engineering. So it's like copying and pasting from chromosomes. So you put recombination sequences in the genome, and then you get the translocation you want. Okay, and you can do random metagenesis with chemicals, that is to explore function, but we are not going into that. So, how do you do that? Well, of a fertilized egg, you take that fertilized egg and then you implant it into sort of pregnant female, and then after that you generate an F1 or offspring. And here in red I'm showing the transgene in the mouse, and some of them will be positive, will have integrated the transgene, some of them not, and then you will select those, and then you will breed them and make line of transgenic mice, and you'll start studying them. And I'm going to show you an example right now. Okay, so you remember I showed you this. This is the pathway. So if you want to make a melanoma, we're going to take a mouse and change one of these, either express it, knock it out, or do something. Okay. So that's what we've done in collaboration with Susie Chen in Rutgers. So we took a gene which is important for melanoma, and we put it under melanoma specific promoter. Then therefore the gene is expressed only in melanocyte, and what happens is that these mice after 4 to 6 months start developing melanoma. You see shown by the dark spots here. This is just a cartoon, and this is the real mouse. See, you see the ear of these mice are black, okay? They are darker, they are thicker, and the tails of these mice are darker and thicker, okay? And if you see here, this bulge here is even a worsening of the melanoma of these mice, okay? So, meaning that maybe they had other mutations, okay? The other events, okay? So, and okay. Just came back from lunch, this is for dessert. You can see here the melanoma is going to the lymph nodes, okay, and you can see here in the skin the melanoma. You can see here the spleen is a little darker, the liver is darker, okay. So now that we have this model at hand, this is the melanoma in the skin, okay. This is a section of the skin, so this is a little bit of melanoma. This is a lot of cells growing, okay. So, but we need to cure the melanoma, so how do we do that? And before doing this, so what we do, remember I told you chemotherapy doesn't work well in melanoma, and radiotherapy doesn't work well with melanoma, so we want to try something new and something different, and that is immunotherapy. So how do we do that? I'm going to just recapitulate a little bit and tell you something very basic in immunology. So everybody knows, I think, or should know, that the immune system is designed as such that if you bring in a pathogen, which is here a bacteria, the immune system will recognize that this thing here is not self. This is non-self. So what's going to happen? So the mouse is sick. The immune response, you'll have an immune response. You'll have T cells, antibodies, whatever. They are going to attack the bacteria. Okay, and then the bacteria will be dead, and the mouse is healthy, okay? But cancer cells are not non-self. So if we compare cancer cells and bacteria, you see the bacteria is a pathogen, it's different from the mouse, it's recognized as non-self, okay? So we will have an immune response. If we take tumor cells, it's the same as the self, so it's not recognized by the immune system, so there is no immune response. So we cannot have an immune response against cancer. So how do we do that? So one way of doing it is the following. So we take a gene that is expressed only in melanoma. So if we take the mouse gene and we inject it to the mouse, as I told you now, it's recognized as self, and we don't have an immune response. So what we do, we take the human gene, which is very similar to the mouse gene, but yet it's different, and then we inject it to the mouse. So the mouse will recognize it as non-self, and we will have an immune response, and therefore we will have